Hello, everyone, and thank you for joining us for this first EMBO Open Colloquium. Before we actually get started, a couple of housekeeping instructions. Anyone here can submit a question at any time. You'll see a Q&A box. You type your question in there, and anyone in the audience can upvote this question. So this will help me figure out which questions to pose to the speaker because we may not be able to cover all questions. Okay, and the um, other point is there is this separate session for students and postdocs to interact with John and Markus right after the second talk, which would be Markus's talk. Okay, so we get started now, and what I'm going to do is introduce the topic before we go on to the main event. The idea behind this particular colloquium and this set of spectacular speakers is to think of metabolism as a central form of biological information flow. So here we want to kind of sweep across a very broad space. Metabolism is an ocean, and here we can think of metabolic networks, biochemical evolution, self-organizing processes that drive complex biological behaviors, biological rhythms, and cell states. So essentially what the idea is that uh, metabolism is at the heart of information flow within cells and between cells. We all know that all cells respond to nutrients and environmental cues and eventually regulate different cellular outcomes. All of those cellular outcomes lead to more complex physiology. But when you think of regulation and when you think of cell fates, oftentimes you're thinking of cell groups, cell functions in the terms of complex transcriptional networks, signaling, proteostasis. What is often kind of taken for granted is that metabolism is a passenger in this process. Actually, in reality, what happens is metabolism is the first thing that responds through a combination of just mass action and thermodynamics, as well as through regulatory processes. So the key thing here is that unlike what a lot of biochemistry courses teach you, metabolism is always dynamic and never static. It isn't just one reaction going on where A is converted to B by some boring enzyme, but it is very dynamic, highly regulated at the level of thermodynamics, mass action, flow, as well as active regulation by regulatory proteins. The other thing to remember is that there's no such thing as a stable equilibrium in biology. There's only an attempt at homeostasis. And and so um, uh, metabolism and all other regulation is a two-way street. It isn't just that signaling or transcription regulates metabolism, but metabolism itself and metabolites directly regulate a whole bunch of regulatory processes in cells. So what we need to fully appreciate this ocean of metabolism is a systems level perspective integrated with its roles in physiology. And that's really the underlying idea behind the speakers. So the key concept when you think of metabolism as a systems level regulatory process is that a cell requires often mutually biochemically incompatible processes, but these are essential for the cell to survive. The second major point is that key metabolites, depending on how much of them are present, can directly control biological outcomes and effectors by directly modifying them as well as by being utilized. So it isn't just that metabolites are being utilized. Regulation has to be tightly coordinated with metabolic states, and these key metabolites, through their sensing signaling systems, affect that coordination. There is actually a reasonable organizational logic to metabolic regulation, which can be derived based on biochemical logic and needs. And that collectively, metabolic processes and metabolism can drive collective behavior as well as emergent phenomenon in biology. So what we're going to kind of see in at least some of these talks is that we don't always have to deal with very complex physiology to learn something fundamental about how metabolism controls more complex biological behavior. For example, we've learned a lot about metabolic organization and cell state from simple cells like yeast or cyanobacteria. And these findings, these conceptual findings, have actually scaled very well across organisms. So a couple of ideas that really came out of work with very simple systems are the idea that the cell cycle itself can be driven through metabolism, and the other idea, which would be the other part of today's talks, is that metabolism can control complex biological rhythms. This is again studies that came out from yeast and from cyanobacteria and so on. In fact, even in our own lab, in my lab, we look at how metabolic constraints determine resource sharing or division of labor or organized specialization in groups of cells, how resources build up in cells that are in a particular environment, and these resources can be produced or shared. And this can allow some other cells to consume them and completely transform their states, their differentiation, their function, their growth. And so you can have stable, organized heterogeneity within complex environments, even when you start off with genetically identical cells, all of which are growing in the same environment. So the idea here is that resources and resource thresholds can control organized heterogeneity, and self-organizing biochemical networks, which can mutually exclude incompatible processes, can drive specialization and division of labor in cells and groups of cells. Groups of cells can then function together for collective growth and fitness. So some of this is based on this underlying deeper idea of what does a biochemical cost mean to a cell, and how important context is when you're thinking about metabolism. So, okay. So now on to the main event, which are two wonderful speakers, John O'Neill and Markus Ralser. And the idea of having both John and Markus here is to kind of connect these distant dots in metabolism and see how metabolic control and organization can control complex behavior. So John's lab is trying to understand the molecular basis of biological timekeeping, of biological rhythms, with two kind of broad areas. What kind of inputs and outputs connect timekeeping with physiology? And more fundamentally, what explains cell autonomous timekeeping, and what are the post-translational or what are the ancestral clocks? And in this in particular, John's lab and John's work in particular has really brought out this idea that a biological clock or a circadian clock need not just be a simple transcriptional feed-forward feedback. The classical idea of a circadian clock is this per clock PMAL kind of a transcriptional network in mammals. You have these transcription factors, they go up and down during the day, and then they control physiology. But early work from John, a spectacular work about 10 years ago, just kind of made you just sit up and gasp, because in these two beautiful papers, John showed two things. First is that circadian rhythms persist without transcription in a eukaryotic cell. This was in an algae. And then even more amazingly, you have robust circadian clocks in human red blood cells, which don't even have transcription. They don't have a nucleus. And since then, John's lab has been doing beautiful work in integrating how metabolism and circadian rhythms, biological rhythms are integrated. So John would go first as the first speaker, and then our next speaker is Markus, whose lab has probably brought out some of the most original ideas at a systems level of how metabolism and metabolites are organized in biochemical networks, and how you could think of biochemical evolution. So, Markus's lab has been breathtakingly productive over the past few years, but two very original ideas are coming out of the lab. One is that you could think of self-establishing communities and how they enable cooperative behavior in a eukaryotic cell, which would then be the basis of cell community formation. And the other really wonderful set of ideas that the lab is exploring is this idea of biochemical evolution. How do biochemical pathways actually come about? Based purely on the rules of biochemistry. So you could think of how a pathway like gluconeogenesis can emerge starting with just precursor metabolites and without even having a complete set of enzymes, or how, say, something like the Krebs cycle could evolve when you're thinking about redox biochemistry. And in all of these studies, which are very different, Markus's work, John's work, very different, you can see these connecting threads of how biochemical logic of processes can fundamentally drive complexity in biology. So, without any further ado, we're going straight into the first talk, which would be John. So, John, if you could share your screen as soon as I exit mine. And the format is John will speak for his 15-ish minutes. We'll have a few questions. You can save some more questions for later in the colloquium. Once John is done, we have a few questions. We'll then transition to Markus. And so, John, it's all you.
Fantastic. Thank you so much for organising this virtual symposium, Sunil. The great thing about you not being able to see me during the introduction is you couldn't see me blushing, and probably Markus blushing when you're saying such beautiful things about our research. So, as Sunil said far more eloquently than I probably will, my lab, and for a long time, I have been interested in the mechanism and function of biological rhythms. Primarily, we are talking about the circadian clock in mammalian cells, and this is present in pretty much every cell of your body. Here, we're looking at a time-lapse imaging of mouse fibroblasts, where the luciferase gene is fused to the period 2 gene, just allowing you to observe these very stable and robust rhythms in clock protein activity over many days. And so what we're really trying to understand is what are the minimal set of biochemical components and their interactions that are necessary and sufficient to explain this 24-hour rhythm, which we basically can't at the moment. Now, recently, we've tried to get some insight from a different type of biological rhythm, the ultradian rhythm in yeast cells in Saccharomyces cerevisiae. Now, this is much shorter than 24 hours. It's usually about a 2 or 3 hour period of oscillation compared to 24 hours or approximately 24 hours that you see in mammalian cells. And in our recent work, we've come to the conclusion that energy efficiency seems to be one of the key reasons that such biological rhythms are found throughout the tree of life. And to understand why, let's start by considering the fundamental requirements that cells have for viability. So, as Sunil said, you need to maintain your proteome because protein activity is essential to every biological process. And cells need to maintain the correct amount of each protein or within the right homeostatic range in the correct compartment to perform its various functions. This housekeeping consumes a large proportion of cellular energy budget because protein synthesis requires an awful lot of ATP, and every protein must be replaced at roughly the same rate that it's degraded. This is particularly true for multimeric protein complexes. They're even more expensive because their subunits have to be assembled stoichiometrically, and this means they need to be synthesized at the same time and in the same place. And so the most efficient way to do this is through a translational burst. You just make a lot of each protein, which consumes ATP at a very high rate, compared with piecemeal assembly, which is obviously going to be less efficient because orphan subunits need to be degraded. Now, cell viability also requires metabolism in order to supply sufficient energetic substrates for protein homeostasis and other essential anabolic and catabolic functions. And also, metabolism needs to ensure that energetic resources are conserved when nutrients are limited. So this primarily occurs through regulation of everybody's favorite kinase at the moment, the target of rapamycin complex, whose activation is prevented when amino acids or energy charge is low. So TORC activation stimulates protein synthesis and other growth processes and liberates the machinery that is required for protein synthesis from ribonuclear protein granules or biomolecular condensates, such as P-bodies and stress granules. Now, something that people tend to forget about after first year physiology lessons is that in order to remain viable, the cell cytosol must be in osmotic equilibrium with the extracellular environment. Otherwise, cells lose control of their volume and most proteins stop working. So to compensate for changes in intracellular osmolyte content when TORC is activated, we think cells export ions such as potassium, which is the primary, most abundant osmolyte in the cell, in order to maintain osmotic equilibrium with the extracellular environment, in order basically to ensure that the cytosolic activity of water remains the same. So we're going to start with what we hope is the simpler model. Like circadian rhythms, these yeast rhythms affects most aspects of cell biology, including mitochondrial respiration. And here the period of oscillation is just a few hours, much shorter than a day. When yeast are grown in one of these bioreactor devices under nutrient-limited conditions, these cell autonomous rhythms synchronize with each other, allowing us to monitor them over many days using an oxygen electrode, for example. Now, a consistent feature of these yeast rhythms is a phase of low oxygen consumption, low respiration, which we'll call LOC for short, when respiration is low or decreasing, followed by a briefer phase of high oxygen consumption when respiration is increasing. Also, like circadian rhythms, these ultradian rhythms are effectively independent of the cell division cycle, but they do affect cell division by preventing DNA synthesis from occurring during HOC. Also, a clear difference with circadian rhythms actually is that the period of these yeast oscillations is very sensitive to nutrient supply, which can be adjusted simply by changing the dilution rate. So as the rate of glucose supply decreases, you can hopefully see that the period of oscillation is getting longer and longer. And with a few simple controls, we can actually turn this dissolved oxygen concentration, which is just a steady state measurement, to a dynamic measurement of oxygen consumption rate and the way that it changes over time. And what we noticed, and hopefully you can see, is that the duration of HOC, which I've color-coded in red, really doesn't change between these three different dilution rates with three different rates of nutrient supply quantified here, whereas the duration of LOC clearly changes as a function of dilution rate. So this suggested to us that LOC is determined by external factors, whereas HOC is cell intrinsic. And this suggested a new way of understanding the oscillation by considering what's happening during each of these two distinct phases. We started with a really simple question because protein synthesis is really horrendously energetically expensive. So perhaps that increase in respiration during HOC is simply attributable to an increase in protein synthesis. And using puromycin incorporation, we get evidence that strongly supports that and coincides with an increase in TORC activity reported by phosphorylation of S6 kinase. And so we see that during HOC, there's a clear increase in protein synthesis and TORC signaling, and this decreases during LOC in blue here. Now, an increase in cytosolic pH is a well-known signal for TORC activation in yeast, and using several different methods, we found that this increase in respiration was accompanied by a parallel increase in proton export, which led to an increase in cytosolic pH by roughly 1 pH unit. So much higher during HOC around pH 7 than during LOC around pH 6. And at the same time that we see this increase in protein synthesis, we see a decrease in free amino acid concentration at all three dilution rates, very much as you'd expect. You know, if you're going to make some protein, you're going to have to use amino acids to do that. Okay, so that's protein homeostasis. And then in order to provide the machinery for protein synthesis stimulated by TOR activation, we observed the liberation of many different proteins required for protein synthesis and metabolic enzymes from biomolecular condensates that again increase during HOC at the same time that we see this increase in OCR and proton export. And obviously it's not just ribosomes and proteasomes and chaperones. We're also talking about known stress granule markers such as PAB1, which becomes much more diffuse during HOC and then much more punctate because it's in RNP granules during LOC. Okay, so what's happening metabolically? So during HOC, with this increase in respiration, you'll notice that there's a precipitous drop in AMP, adenosine monophosphate, which is a very reliable proxy for energy charge in the cell. So what it's saying is that there's an awful lot more energy available to the cell during HOC compared with LOC. And at the same time, we see this profound drop in the abundance of storage carbohydrates such as trehalose and particularly glycogen. So just like mammalian cells, yeast store most carbohydrate in the form of glycogen, and the abundance of glycogen or carbohydrate stored in glycogen changes by about twofold over the course of the oscillation, accumulating during LOC and then being consumed, being liquidated during HOC. And this is because glycogen is simply providing the fuel for this increased mitochondrial ATP generation, which is in turn being consumed by an increase in protein synthesis. So as the concentration of cytosolic protein increases during HOC, the cell obviously can't swell because it's got a cell wall there. So to maintain osmotic equilibrium, it exports potassium and other organic osmolytes such as betaine during HOC. So they crash down as cytosolic protein increases, and then they gradually accumulate again during LOC. So I just bombarded you with a lot of information. So here's a nice little summary diagram that during LOC, we see sequestration of the protein synthesis machinery, proteasomes, ribosomes, chaperones, metabolic enzymes into RNP granules, biomolecular condensates. And during LOC, we've got an accumulation of osmolytes, amino acids, stored carbohydrates, but respiration is low and pH is low. And then at some point, there is a switch which is flipped and the cell goes into this shorter burst of high oxygen consumption, where all of these stores are depleted rapidly with an increase in respiration and translation and an increase in pH. So overall, this is consistent with a model where oscillations occur simply because nutrient supply is not sufficient to satisfy both cell growth and efficient protein synthesis at the same time. So cells initially sequester and store metabolic resources, accumulating polysaccharides, accumulating amino acids and osmolytes via increased transport, autophagy, and biomolecular condensation. And this is all maintained by low cytosolic pH. Replete stores trigger an increase in proton export, which elevates cytosolic pH, stimulating TORC1 and liberating the protein synthesis machinery from RNP granules. This facilitates bursts of increased protein synthesis, the liquidation of carbohydrate stores, which sustains higher respiration rates and increased ATP turnover, with the export of osmolytes being absolutely required to maintain osmotic homeostasis. And then HOC ends because either your carbohydrate or amino acids have run out, or because the rate of ATP synthesis is just not enough to sustain protein synthesis, or due to an osmotic challenge. Now, because the duration of translational bursting is invariant and determined entirely by cell intrinsic factors, the period of the oscillation is determined entirely by the time that cells take to store adequate resources during LOC in order to license passage through this pH-dependent metabolic checkpoint that initiates translational bursting. Of course, there's an awful lot more detail and supporting evidence that I'm skipping over here. So I do advise you to go and read the paper if you want to know more, particularly the supplementary information. All the important data is in supplementary figures. But the upshot is that this mechanism not only explains rhythm generation and frequency. So period is determined simply by the time taken to store adequate resources, but it also tells us about the function of the oscillation, that rhythms minimize energy expenditure by restricting protein synthesis until sufficient resources have been stored whilst maintaining osmotic homeostasis and protein quality control. So according to our model, inhibition of translation or torque during HOC should bring that increase in respiration and the increase in proton export crashing down to the same level as LOC, because you've removed the drive for that increase in respiration and proton export. And so that's what we observe. You treat cells with rapamycin, you no longer see this increase in respiration that would normally occur during HOC, and exactly the same thing when we treat cells with cyclohexamides to inhibit protein synthesis. So the level of respiration is not going below the basal levels that we see during LOC, it's just not increasing because that HOC increase is driven by increased protein synthesis. And similarly, if our model is correct, then you would expect that cells which can't store carbohydrates shouldn't be able to sustain rhythms and should show a deficit in protein homeostasis and quality control. And so that's what we observe with glycogen synthase and phosphorylase mutants. Cells which can't mobilize or store carbohydrates as glycogen, they don't show rhythms. And then we see an obvious deficiency in protein quality control in that you've got about 50% more protein aggregation in these glycogen storage mutants compared with wild-type controls. And then finally, the most complex prediction we tested is that potassium depletion will shorten the period of oscillation because it's going to lead to a truncated HOC because it can't buffer intracellular osmolarity, but also a truncated LOC because the stores, the carbohydrate and amino acid stores, weren't consumed during the preceding HOC. And so that's what we observe, that as we wash out potassium extracellularly, cells flip into this very, very short period of oscillation in a dose-dependent fashion. So dose-defined or period defined by the concentration of extracellular potassium. So what we're saying basically is that nutrients, when they're limited, translational bursts are the most efficient way to maintain protein homeostasis. And thus metabolic rhythms in this case are the inevitable consequence of rhythms in protein synthesis, which confers a selective advantage under nutrient limited conditions. So the only way to efficiently produce protein complexes in particular is to store glucose as glycogen until stores are full, and then you get a spillover leading to increased ATP synthesis, increased proton export, activation of torque, which changes the biophysical characteristics of the cytosol, liberating protein synthesis machinery and metabolic enzymes, and allowing this dramatic increase in protein synthesis rate. Now, going back to mammalian circadian rhythms, there is good evidence that the timing mechanism that drives these yeast rhythms shares a common origin with the daily clock that drives circadian rhythms in mammalian cells. For example, casein kinase regulates circadian period in all eukaryotes, including mammalian cells. And although both biological rhythms are accompanied by changes in transcription, this is almost definitely a consequence of rhythms in metabolism and not the cause, because very few proteins actually show functionally significant oscillations in abundance. And this is really much more consistent with the idea that biological rhythms really function to maintain protein homeostasis over time, rather than to make rhythms in the levels of proteins, which would, of course, be energetically wasteful. In both circadian and ultradian rhythms, we observe oscillations in torque activity that drive rhythms in protein synthesis and cytosolic crowding, supported by dynamic and adaptive changes in cellular metabolism and ion transport. So here's just an example of that, a rhythm in soluble protein, but not total, in mammalian fibroblasts, accompanied by a rhythm in torque activity and translation. Again, there's no change in cell volume, and so cells need to compensate for that increase in cytosolic osmotic potential by exporting potassium, sodium, and chloride, when protein levels increase and the converse when protein levels decrease. And then just like in yeast, we see now a daily variation in respiration and glycolytic flux. But then one critical difference is that circadian rhythms are metabolically compensated, meaning that the period of oscillation is effectively insensitive to extracellular nutrient availability. And this highlights a more general point that whilst there are clear similarities, the two different cellular rhythms must be generated by different mechanisms, because not only this difference in metabolic sensitivity, but in mammalian cells, torque activity is not essential for rhythmicity, whereas in yeast cells it is. And then there's an obvious difference in secondary active transport, and also mammalian cells don't use pH as a second messenger. So in yeast cells, we have a testable and predictive model that accounts for the oscillation and its period, where central metabolism, signal transduction, active transport, and the synthesis machinery are coordinated to meet the bioenergetic demands of efficient translation. This leads to the hypothesis that oscillations in protein synthesis rates may confer a general fitness advantage under nutrient-limited conditions simply by minimizing the cost of protein complex assembly. In mammalian cells, we still don't fully understand the timing mechanism, but we do observe similarly organized rhythms of torque activity, translation, metabolism, ion transport, and the protein synthesis machinery to that which we observe in yeast, which strongly suggests a similar function for the circadian rhythm as we've shown in yeast, leading to the hypothesis that we're now testing that circadian rhythms allow daily consolidation of proteome renewal, facilitating homeostatic regulation of protein complex levels whilst minimizing their energetic costs. So you really want to maintain the activity of protein complexes at the same level, and it's most efficient to turn them over, to remove them and replace them all at the same biological time of day when the most energetic resources are available. And so thanks so much for your time. I'm sorry if I overran a little bit. These are my collaborators and wonderful lab members, both present and past. And I think we've got time for a few questions, and then it's Markus. So here
Fantastic. I am. Thank you, John. This was a great talk. So what we're going to do is maybe have about 3.5 questions. I'll just read them out so that the audience can listen to it. First question from Isabel Lopez Maya is: how sensitive are LOC-HOC oscillations to temperature, and are they sensitive to nutrients other than glucose?
Oh, that's a really great question. Okay, so the oscillations are, so LOC and HOC are sensitive to an acute change in temperature. And that is largely because translation itself is temperature sensitive. So if you apply an acute temperature increase, you will get a truncated HOC because translation will speed up. However, there will be a commensurate change over the length, over the duration of an entire cycle. There will be a commensurate shortening of LOC. And so what we find for these oscillations is just like with circadian rhythms, overall they are temperature compensated. So the period of the oscillation does not change at different biological temperatures that yeast can tolerate, but you do see acute responses to temperature shift. And then, yeah, you can get these oscillations when other nutrients are limited instead of glucose. So they've been reported to run on ethanol as well, although there's only a couple of papers on that. And there, I think the argument would be that rather than the focus being on glycogen per se, the shift is, there's probably a shift to differential fatty acid metabolism. But as I say, there's only a couple of reports on that, so it's something that would really need to be followed up before you could be particularly confident about it.
Okay. Kind of two questions merged into one. How efficient is the LOC-HOC switch to rapidly changing environments? And if the oscillations are responses to chemical changes, how are they affected by physical cues if you have mechanical forces or adhesion?
So mechanical forces, I've got absolutely no idea, and I've got no I can't immediately think of any case where anyone has tested that. But certainly the oscillation is quite sensitive to external acute perturbation. So Ben, too, when he was working with Steve McKnight, published several papers showing that you can shift the oscillation by providing a bolus of glucose, which shifts into HOC, or if you provide a bolus of acetate, which also shifts into HOC. So basically you're providing more metabolic substrates, and so you can kind of overcome the need to fill up your glycogen stores. And then there are reports that hydrogen peroxide also leads to a elongated LOC, as I recall, and also hydrogen sulfide. I can't remember the exact effect that hydrogen sulfide has, I'm afraid. But yeah, there are a number of external perturbations that you can add, which acutely shifts the phase of the oscillation. And in fact, there's a really nice paper just came out in Nature Chemical Biology from a guy, Bruce Dickinson's lab. And there they really drill down into the basis of this phase shifting in response to changes in internal redox state. It's a very, very nice bit of work, which I'd recommend you have a look at.
Okay, this is another question from Priya Crosby. If I remember correctly, the YRO only occurs in fairly tight nutrient environments. If the nutrients are in excess, do you see such a temporal segregation in protein production and carbohydrate consumption, or does this disappear?
So that's a really great question. I can't provide a conclusive answer because no one's done the experiment yet. When nutrients are in excess, you no longer observe oscillations at the population level. So these type of measurements that we've been doing become impossible, and you have to do single cell measurements. Now, there's a lovely paper, Papagiannakis et al. In Molecular Cell 2017, I think, where they demonstrate that these oscillations continue at the single cell level when all nutrients are in plentiful supply, but they did not look at protein synthesis. However, the implication from what they've shown so far is that these rhythms in protein synthesis persist at the single cell level, but they desynchronize from each other between different cells in the population. So what you'd really need to do is some single cell protein mass spec, I guess, in order to demonstrate that there are still coupled rhythms in protein synthesis and respiration when glucose is in excess.
Okay, we do have more questions, but in the interest of time, we're going to save them. And if you guys who have questions join us in the open session later on after Markus's talk, we can have some of these questions. Okay.
Brilliant. Thanks,
so thank you, John, for a fantastic talk. And now we're going to move on to Markus Ralser, who's going to talk about something quite different and equally interesting.
Thank you so much, Sunil, for the nice introduction before, and thanks so much to Gerlind and the EMBO team for organizing this. So let me try to share my screen. I think that would be a good start now for this seminar. So I hope it works. So when preparing for the seminar, I realized that I'm affiliated now with the EMBO family more or less exactly since 10 years when I became a young investigator. So I digged out a little picture from the time, which has been created by one of my former PhD students and illustrates a little bit what we are after today. So after John's talk, I continue a little bit with yeast. This is not everything we do, but Sunil asked me to specifically give a little bit of conceptual talk in the seminar today to introduce some concepts, and we use yeast a lot for systematic studies into explaining how metabolism functions and how it interacts. Now, the key problem we're gonna address has to do with the central dogma of molecular biology, which was introduced by Watson and Crick, believe it or not, already in the 60s. So 50 to 60 years ago, we came to know how the main flow of information is happening in biological systems. So you start. Can you see my screen, Sunil? Does it work? My screen doesn't.
Oh yeah, no, it's very clear, no problem.
And you see my pointer as well.
Yes.
Fantastic. Okay, great. So Watson and Crick implied this flow of information as we have it now, starting from a genome and having then self-organization, having input from outside, having metabolic input, and this would eventually create the phenotype of an organism. So why is this so important? Of course, if we understand how we move from the genome to the phenotype, we understand how biological processes work together. And so it's good for basic science, but there are also a lot of implications and applications in real life. So one of them, if you could do this, you could sequence a genome and you would perhaps know if a certain individual would respond in a certain way to a drug. Or in biotechnology, if you would understand this, you would know how to manipulate an organism to produce whatever you like. So there are a lot of implications if you understand the flow of information from the genome to the phenotype. The problem is, it isn't that simple. No, we now know about this concept since 6 decades, and still you cannot sequence a genome and give it to somebody, and this somebody would tell you what's the phenotype of that particular cell in that particular environment. So why is it so complicated? Well, there are several reasons, of course, for this, but one of them is it's probably not that linear, as illustrated here in this toy graph that I stole very nicely from the Khan Academy, but also because we have not speaking that much about these little bystanders here, as Sunil called them in the introduction, about metabolites. So metabolites play a role in each of those steps, going from the genome to the transcriptome, to the proteome, to the phenotype. But they are not just there to make the building blocks, they integrate with each of those steps and they regulate it. And that's in fact where it becomes complicated. Now, so on this graph here, I'm not sure if you see this control thing here, but anyway, you need to believe with what's behind it if you see this. So this here is a simple experiment that illustrates this. So these are 97 yeast knockout strains, not so important for the time being what they are, and they have been grown in the same flask. And one of my talented postdocs took a sample out of the same flask and measured a very precise quantitative proteome and a precise quantitative metabolome from the same flask. So no variation between the two. And then you compare what you get. And this is a very simple hierarchical cluster. And what you see at first instance is that the proteome gives a cluster and the metabolome gives a cluster. And if you compare the two clusters, you see a not significant cophenetic correlation. So if you look at this at the first place, you get the feeling that the metabolome would have nothing to do with the proteome. So it tells you a completely different picture about the same strain. Now, if you think about it, with all the knowledge you have about biological systems, you realize that can't be true. The reason being, the proteome and the metabolome are highly interconnected. It goes in both ways, but effectively, the enzymes that are quantified as part of the proteome, they synthesize the metabolites, and the abundance of those enzymes to a certain degree depends on how metabolism is functioning. Okay, so there is clear connectivity between the proteome and the metabolome, but you are not capturing it with a simple linear regression, with a simple linear mapping. So why is this the case? The case has to do because the structure of the metabolic network. Now, does this flip here? So this is now an illustration of metabolism. I have stolen this one from the Keck database, but I know that many of you have seen very different metabolic maps. So the most scary ones are these Bollinger charts that we used to pin on our walls in the 1990s. Okay, but what can you see? But the simplest thing you see is that you have not a one-to-one relationship between metabolites and enzymes. You have a many-to-many relationship. Yeah. So many of those metabolites, they bind to many of the proteins, and many of the proteins have different metabolites as substrates. Now, so there is not a one-to-one dependency. That's the first thing. Second thing is that metabolites are in a constant flux. So you take, let's say, here a glucose into the cell, you convert it through glycolysis, you convert it in the TCA cycle, you make an amino acid out of it. Yeah. And so if you manipulate an enzyme here, depending on the activity of the enzyme and depending on the speed of the reaction, the biggest concentration change might be here. So if you now try to correlate this enzyme with this reaction here, just picked out a random one, you may find that the connectivity is not represented in a simple linear correlation. Okay, so there's a many-to-many relationship. And that's one of the reasons that we get confused in predicting the phenotype the moment we leave the proteome and do the jump to the metabolome. So, and this is one of the things my lab addresses the most. We try to use systematic experiments to derive the logic of this network, to see which rules are behind it and how we do it. Okay, now I'm focusing a little bit on these perturbation experiments because I believe they illustrate quite nicely how metabolism is functioning, how it does. Okay, when we started in that field, we said, well, the best way to get there is whether we have a collection of different gene knockouts, because they're clean, and connect them to the metabolic phenotype those gene knockouts create. And in this way, we would be able to map metabolic changes to changes in the genome directly. So in order to do that, we had to do some work. So the first problem we had is that yeast as a model organism was heavily manipulated to be used in the lab. And one of the manipulations that has been done by our scientific grandfathers is they have deleted some essential metabolic genes in those yeast. So yeast are fine with that. They grow in rich nutrient media very well. And those metabolic genes are totally useful for doing genetics. The problem is, though, that microbes, all of them, also higher organism cells, they're very, very lazy when it comes to metabolism. That means the moment you give nutrients to the cells from outside, they would shut off their own metabolism and uptake all the nutrients. So if you do an experiment in a rich media, you effectively measure in a metabolome what the cells have taken up from outside, and you are not seeing what they would synthesize. So in order to look into biosynthetic metabolism, the first thing we had to do is to fix those genome scale collections of yeast. We reintroduced over mini chromosomes all of the missing genes, and we created a library of about 5,500 yeast strains, where each strain has another gene deleted. It has an intact metabolic background, and where we can do systematic metabolomics with it. Yeah? Another part of the work was to establish new protocols that enable us to grow those cells not in small flasks, but in multi-well plates in very high throughput, and that we have ways to use pipetting robots or liquid handling systems to extract metabolites and systematically create metabolite extracts. I come later a little bit to what we do with the proteome. And eventually we had to work on the analytics to bring analytics to scales that we could measure thousands of different samples. So the first experiment we then started to do on a genome-wide scale is to look at amino acids. So by amino acids, they're very important metabolites. So they converge many different parts of metabolism into a small set of metabolites. So you get a good overview. Second, because of these deficiencies in amino acid pathways, we believe this is the area where people might have missed the most of the knowledge. And third thing, and that's not that unimportant, we can measure those very precisely. So for all of you that ever scale up an experiment, which you initially did with a few strains to thousands of them, you need to be very careful about measurement precision. Yeah, because noise becomes a big issue, batch effects become a big issue. So you need to be much more careful in doing a system scale experiment as you need to be when you do a one-to-one experiment in comparing, let's say, a wild type with a knockout. Yeah. So we made a method. It's based on multiple reaction monitoring. It's very sensitive, it's very precise, and it quantifies 19 of the 20 proteinogenic amino acids in about 2.5 minutes of time. Okay. Great. And so what we did with the tool is we created this huge matrix which connects now every single gene deletion that is non-essential for the yeast genome to the metabolic signature. Okay, what can you do with things like this? So the first thing is you can get a system scale picture and see, okay, how many genes do actually influence the biosynthetic metabolome? And the answer was not 1 and 2, but it's at least 1/3 of the genome. It's probably much more if you would take into account multiple growth conditions. So a huge fraction of the genome is influencing directly or indirectly metabolism. Then you can do gene set enrichments to see what are now the most enriched players. Then you find things you have expected, like the mitochondrion, so metabolically active organelles which drive it. Then you see a lot of things which have to do protein biosynthesis with ribosomes and translation. So this is again this feedback principle. So those processes which consume most of the metabolites that need them, at the same time the sensors which tell the cell how much to make of them. Okay, there is this clear connection. And one thing which surprised me a little initially is that we didn't find many clear transcription factors or protein kinases, specific ones that would have broad impact on metabolism, but we found a huge enrichment of everything which has to do with the chromatin, with histone modifications, with things which play at the big scale. And we now believe this has evolutionary reasons because the regulation of metabolism had to be something in place very early on in evolution. And so many of this general transcriptional regulation probably was there initially to control metabolism and vice versa. Okay, so allowing the cell to react to different changes in the metabolic environment. So that's the first thing you can do, you find the genes. But you can do other things, and one of the things which you see slightly on this heat map, I'm not going into detail, but you can cluster those metabolomes. And then you cluster genes where you didn't know which function they have. You can cluster them together with genes where you know the function, and in this way provide a functional association to the so far unknown part of the genome. And we were the first ones who did gene annotation on a system scale via the metabolome. And it turns out, because of that, it's highly informative. And this first big experiment annotated 1/3 of the at that point unknown yeast genome and assigned functions to the genes. Yeah? So metabolism is a lot informative in both ways, not only how metabolism responds, but also metabolic profiles tell you what all those genes in the genome can do. Okay, so why is this so important? It is important because even that small part of metabolism that we have been looking here, the amino acid metabolome, has multiple functions in the cells. Yeah? It's not only there to make proteins, although this obviously is a key and important reason, but those metabolites are implicated in quite a few different processes. And I picked up one, which I still believe is a nice example, which shows what you need those understanding of those metabolic processes for. So in studying amino acids, we noticed a weird behavior initially, which we couldn't explain, and we later named it lysine harvesting. So what's lysine harvesting? So that's the lysine concentration in a wild type yeast that grows at maximum speed. Okay, it has everything it needs. It's a happy cell. It grows very fast. And it synthesizes the amount of lysine it needs for growing. And that's the concentration of it. Now, if you give the same cell lysine from outside the cell, yeah, it will not take up just the amount of lysine it needs. It takes up about 100 times more lysine from the environment, at least in a concentration level, compared to what's required for growth. And we initially didn't know whether that's active or passive, but simple explanation, we found some genes which are required for this excess uptake. So we believed initially on, well, this is adaptive. This is not by random chance. Okay, so this was counterintuitive. So why is this counterintuitive? Because we have realized before that metabolic networks work the most efficiently if they have a minimal concentration of each of its metabolites. And the reason is the more metabolites you have, the more interferences you have between biochemical reactions. So high metabolite concentrations can be really bad for metabolism. So cells typically try to have as little as a concentration of a metabolite as possible, but we saw here that voluntarily those yeasts uptake 100 times more lysine than needed. So we thought, okay, what's the explanation for it? And the explanation came from something where Sunil is now very active in the field, and this is that metabolism is not only occurring inside the cell, but it's clearly very important what happens between cells. That's important for higher organism cells a lot, but it's equally important for microbes. It was probably a little bit anticipated before, but they always interact metabolically. So what we do a lot to study this behavior is we create synthetic yeast communities, and there is one of those models that we call self-establishing communities. So this here is a picture of such a self-establishing community. It's in that case a 2-day-old yeast colony. Picture has been done from below, and the different fluorophores indicate cells of different metabolic capacity, and they all supply metabolites to each other, and so all together they can work then and establish a communal metabolism, which then enables them to grow on a media where each of those cells on their own couldn't. Yeah? So it's cooperative behavior which enables the cells to grow. Now, what happens here is that those cells, the different colors, the different patches, they have different functions, different metabolic functions, and so they diverge phenotypically because they have a different metabolism. If you do something to those cells, they would respond differently. And this gave us the idea to look whether those harvesting phenomena could be required for some phenotypes, and we found that in that case, the phenotype is stress protection. So just 2 examples. So this is the glutathione concentration in comparing a wild type cell with another wild type cell that is a lysine harvester. And what you see is that the glutathione levels go up by a factor of 10. So that's a lot because glutathione is already one of the highest concentrated metabolites or small peptides in a wild type cell, and its function is to protect them against oxidative imbalances, mostly at least. Yeah? So you see that the harvesting enables the cells to maintain a much higher level of reduced glutathione, and at the same time they become stress protected. So that's an example. That's the wild type cell. If you expose them to a certain concentration of an oxidant, it stops growing and doesn't form any new biomass anymore. But if you do the same experiment with the harvester, you see that it's much more tolerant against the oxidant. Yeah? So this exchange of metabolites that can help cells a lot to deal with environment. And so if you have a natural community, if all of those cells work together, each of them gets a different metabolic function. And so they are protected as a community against changes in the environment and how they apply to cells. Okay, now for the last few slides, and tell you a little bit now how we are going to understand those phenotypes. So I spoke a lot about metabolomes because they're very close to the phenotype, and that's eventually what we need to understand. But one of the problems of the metabolomes is that they're very hard to interpret. So very often if you do something to your cell, growth rate changes and hundreds of metabolites change in concentration as well. It's very hard to interpret. So we learned over time that sometimes if you work on metabolism, you can learn more by looking at the enzymes and the proteins. So the method we had a few years ago was called microflow swath. I'm not going into details, but it suffices to say that this was not a popular proteomic method at the time because it didn't go very deep into the proteome. So you could quantify a couple of hundred proteins, but it was much more high throughput. It could do one proteome in half an hour. And so with this method at hand, we could start a few years back to do not one or two proteomes, but do hundreds of them. So a little bit the philosophy of our lab to do everything systematically, okay? And so we were lucky in metabolism because quite a few of the enzymes we have in metabolism, they are high abundant, and more important than that, they're globular, yeah? And so with this top 800 yeast proteins, we actually covered about 75% of the active metabolic reactions in yeast. And this was nice because of the higher throughput, we now could systematically perturb metabolism not only at the metabolome level, but also at the proteome level, and then see how the cell responds. Okay, now, cut a long story short, suddenly we hit the wall, and the wall was, how can we now link actually the metabolome to the proteome? This goes back to now to the hierarchical cluster that I showed you at the start of my talk. So very often a simple correlation doesn't suffice. Yeah? So how do we do this? And Alexei, the first machine learner that joined my lab, he cracked the problem. So the trick actually, it's pretty simple if you know it. So we use the structure of the metabolic network to link the metabolome and the proteome. So how does this work? So we, as illustrative example, I choose one metabolite that you all know, ADP, and we go back to our metabolic map and we see how is ADP connected. And in case of ADP, it's connected to about 50 enzymes that we quantify, okay? And so then we quantify the concentration of ADP in, let's say, a couple of hundred yeast strains. And in the same yeast strains, we quantify all of those 50 enzymes. And then we start with multiple linear regression, and we link a concentration change in the enzyme to a concentration change in the metabolite. Yeah? And after a while, it turns out what are the key enzymes which now determine through an abundance change, a concentration in the metabolite. So in this case, it's about 10 enzymes that are the bluely colored ones. And then we can use this to either train linear models, but meanwhile, we do much more sophisticated machine learning models, which go to the first order, second order, and third order. And they, because of being topologically imprinted on the real metabolism, can then be used and predict the metabolome out of the proteome. And this is an example now from kinase knockouts. Is all of these kinases there. We measure the proteome, we measure the metabolome, and we try to predict then the metabolome. And this is a comparison now of the predicted metabolome from the measured metabolome and on the whole scale of the cell. And what you see is that we get quite a decent correlation between it. So which means we, to a certain degree, can now derive the metabolome of a cell from its proteome. So R square of 0.6 looks awful, but it's actually pretty good because metabolism is not only controlled over enzyme abundance, it's also controlled over post-translational modifications and changes in enzyme activity and localizations that may be wrong. But this picture actually gives you a feeling that if you look at the whole scale of the network, about 60% of metabolite concentrations are actually driven by changes in the abundance of the enzymes that you can measure. Okay, now, this was machine learning. This was a small set of samples. Ever since we think, okay, now to really do this, we need to go to big scale. We can do a couple of thousand metabolomes, but we only could do a few dozens, maximum a few hundreds of proteomes. And we started to thinking, can we not make this technology better? So we have developed since a lot of different tricks. Some depend on what concern the sample preparation, how you prepare proteomic samples, and some concern the extraction. So we've done a lot of work there to make proteomics systematic. So we've done a lot of work here on the mass spec side. I come to this in a second. And we have done a lot of work on software, which enables us to make proteomics faster and more efficient. And the key part of our platform is a new proteomic acquisition technology that we just released. It's called scanning swath. So what does scanning swath do? Scanning swath replaces, for those who know proteomics, the first part of a proteomic workflow, where you typically select a certain peptide and then you fragment it and you identify and quantify it. So what we do here, we have a scanning quadrupole in the first part of the mass spec, which continuously scans to the whole mass range and records everything which comes out of out of that on a very fast to analyze. Okay, so what's the advantage of it? It's two things. So first, it's very fast, so we can now do duty cycles of a couple of 100 milliseconds, so we can move to very, very fast chromatography. Second thing we can now do, we can assign the molecules as they fly into the quadrupole to the fragments they generate on the fly. The reason is that these scans create a new dimension. It's a fourth-dimension mass spec data. We call it a scanning dimension. And so once a precursor is hit by this window, it zooms through, it records a chromatogram, and the same is true for the fragments. And so we have written deep neural network-based software which can make use of this information to deconvolute complicated spectra. So long story short, what it enables you to do is to do proteomic experiments, which in the past have taken an hour or 2 or 10 hours in the minute scale, even though to seconds. So this is now a faster proteomic method we have, and it precisely quantifies about 1,500 proteins in just 30 seconds. So think about how long it takes you to do a Western blot. Or if we take a little bit more time, like 5 minutes, we can now comfortably quantify 4,000, 5,000 protein groups in parallel. That's nice because this covers metabolism quite efficiently, and we can do lots of different things. Just one little teaser before I need to start talking. We now can go back now to our yeast gene deletion collection, take out our 5,000 metabolically competent yeast strains, and now process them over our platform. And we have done this with a slightly long gradient, so we quantify about 3,000 proteins in all of those 5,000 deletions. And that's now a super nice resource for us, because for the first time in one organism, we now have everything. We have every gene deleted. We know which gene is deleted, so we have the information of the genome. We have the metabolome, we have the metabolic network, and on top of it, now we have a proteome for every single yeast strain, and we can derive the logic of metabolism. And we can now look causally into how are the different processes putting together. And just to give you one little graph here, this is now a genome scale causality network, where we look on which process, which protein do we perturb in the cell, and we look which are the metabolic and the general consequences to the physiology of the cell in a causal relationship. And what we found very positive for me is that actually the key response mechanism from everything you can possibly perturb in a cell is the biggest group of response is indeed in amino acid metabolism. So this now finally gives us the scientific rationale of our very first screen that we have started many years ago looking at amino acids. So we were lucky. Yeah, it worked so well because indeed it's the amino acids who respond the most frequently. And it's a lot of growth related processes, of course, that are also important. Ribosome translation, nucleobase forming processes are key drivers, but you have a lot of specific processes that respond to a lot of specific stimuli, and we are now getting into this, and hopefully we can provide such information also to the whole community to see how things working together in the cell. Yeah, last but not least, I need to thank a wonderful team in my lab that over the years has done all of this. Just to highlight a few of the key people. So Alexei Seleshniak, mentioned him briefly, he was the machine learner. He's now a professor in Sweden and develops very cool artificial intelligence, protein structures, very cool stuff. And Christoph is a analytical chemist. He's now moving to a company who does big scale proteomics. And Vadim is a postdoc who is behind our data deconvolution software. So he is now starting a junior group in working into computational proteomics. And Michael is now running a proteomics facility. He was the person who was behind the amino acid metabolomes. And Maria, she's from Mexico. Now she leads a lab at the National Metabolism Institute in Mexico City now. And Maria is the person behind the lysine harvesting story. So thanks so much for staying with me for all the time. And hopefully there are some questions that I might be able to answer.
It was super. Thank you, Markus. So just a couple of questions before we, for the sake of time, before we go to the other session. One question from Pascal Zimmerman: is the antioxidant effect specific for lysine? How do you kind of particularly explain the lysine phenomenon with glutathione?
There's several things coming together, but the main thing is, if you synthesize lysine, you need a lot of electrons, you need a lot of reducing equivalents, which typically come over NADPH. So you need 3 molecules of NADPH to make 1 molecule of lysine. So the moment your feedback inhibit lysine for a long time, because you store so much, and you free your pentose phosphate pathway and the other NADPH producing mechanisms in the cell for reducing equivalents for glutathione synthesis. And that's why those cells which don't need to synthesize lysine, they can maintain a higher reduced glutathione pool, higher concentrations of glutathione. That's why they deal much better.
Fantastic. And as another question, in your stably established communities, this is Robert Arkowitz. He's asked, in your stable metabolic communities, I assume it's the give and take of metabolites which is critical for the establishment. Is this also the case, as in this kind of give and take is also the case when you establish these stable metabolic communities?
Okay, so this is more complicated than it seems. So we start those communities from a single cell, which is perturbed in 3 or 4 metabolic pathways, and they stochastically lose the metabolic capacity in those 4 over a plasmid which is in there, and then it's lost. Yeah. So, but what we know is that once a cell loses the plasmid, so it becomes an auxotroph, it reconfigures not only the metabolic pathway that is perturbed, it actually reconfigures a lot of other metabolic pathways which are coupled to the bone. So what we see, huge metabolic changes in all of this, and they affect the endometabolome, but they also affect the exometabolome. So effectively what you trigger is an exchange not of only the one metabolite that you have been genetically monitoring. We see changes in lots of different exchange rates. Yeah? So the whole exchange activity of the community gets boosted. And with this trick, they don't start sharing anything which they wouldn't share otherwise, but they share more of it. And that's the trick that we exploit here. And to add something, those communities, they are dynamic. They're never super stable. So they end up, you have a certain equilibrium between the genotypes, but if you follow them over time, you see that's a completely dynamic system where they interact and change interactions, and it's quite fun to study it. So Rob has very cool microscopy in France. So hopefully we can send you at some point some of those communities to follow them.
Okay, fantastic. This one's a nice question. Chao San asks, do short peptides belong to the metabolome or the proteome? So it's actually quite a nice deep question.
Well, both, no? So I mean, in the end of the day, you have a gray zone, right? A gray zone with every biological process, and at some point the protein ends and the metabolite starts. So you can do a synthetic definition and say everything which went over the ribosome is a protein. So it's a very working model, but function-wise, you have short peptides, and you could effectively say, well, some of them are more closely resembling the function of a metabolite than of a protein.
Okay, one last question for the sake of time. Gana Panasyuk asks, I'm thinking about the same metabolite that has different roles in different compartments. So for example, acetyl-CoA in epigenetics and in fatty acid synthesis. So how can we study metabolism compartmentalization by combining proteomics? This is perfect for you.
Yeah, so the thing is, proteomics is at the moment not very good yet at compartment-specific proteomics. Now, there are tricks. You can do this. Metabolomics is even worse. So for metabolome to have very good localization information is quite tricky. So we ended up doing a few studies, not many, where we took a little bit the proteome as a proxy and said if a certain pathway is present in a certain organelle, we take the assumption that the metabolite is also there. Yeah, but this, of course, is not capturing those metabolites which are part of multiple pathways, which are quite a few. And so this is an area where is very exciting developments happening. Nothing is super good yet, but there is a lot of excitement about subcellular metabolomics and linking it to proteomics. And that's an area where we hopefully see a lot in the future. The only light at the end of the tunnel at the moment is in very simple microbes, which also have some compartmentalizations, but at least they don't have membrane-based compartmentalization. And there's a little bit more that people know. Don't know if this is the answer you wanted to hear, but that's a very active field at the moment to think about how does in the cell all of this play together. It's very important, very important.
Okay, that was just fantastic. So what we're going to do, thank you so much, Markus, for a super talk. So what we're going to do now is, of course, thank you everyone for attending. This was a lovely active session with lots of questions. We still have some unanswered ones. Thanks for attending. We will now move on to the closed session that's open for all students over here. So those of you who are not students or postdocs, we'd request you to leave. Thanks for attending. And just a reminder, the next EMBO e-talk will take place on June 9th on predatory bacteria.